Hi, this is Roger Green, host of the podcast still known as Surfing the NASH Tsunami. Today we are offering 6 conversations from episode 41, our review of last week's FDA workshop on NITs, with 2 conversations from each of our 3 interviews with key participants in the event. I start this conversation by discussing the idea of loss aversion, that an individual's decision does not reflect an overall fear of risk, but a specific fear of risking something he or she does not want to lose. Researchers and advocates define loss in terms of time to bring a drug to market or screen fail rates, but for government regulators, loss might refer to prestige or credibility associated with what are publicized as wrong decisions. Naim registers this issue, but notes that in his clinic, no more than 5% of people test in a way that might qualify them for clinical trials, and probably 1 to 2% ever participate. And, he notes, this is in a leading hepatology-only clinic. He contrasts the high percentage population with identifiable NASH, which might be 15%, with the 1 in 1,000 unicorns, as he calls them, in the population that can qualify for trials, and suggests this demonstrates how strongly we need to reconsider testing standards and protocols. Jörn Schattenberg notes that the FDA commentator in the final session suggested we could avoid surrogates altogether and instead do a 1 million-person trial looking for outcomes. Jörn feels this is not feasible. Naim agrees. From here, we move to another issue, the cause of death among patients with F0 to F3 fibrosis not being liver disease. As the conversation continues, Louise Campbell notes this to comment on the idea of doing broader multi-organ or multi-outcome trials with drugs, as have been done with vitamins, and ends her comment by stating, and I quote, "I would rather go into the future making the wrong decisions for the right reasons than the right decisions for the wrong reasons," which represents to her a broader look at how we approve NASH drugs. Naim and Jörn both agree. In the last part of the conversation, I asked Naim what he anticipates FDA might do next. He talks about studies generating more data with more NITs, which might lead to what he describes as his, and I quote, "creed, continuing the biopsy pathway, but simultaneously providing an alternative pathway based on using combinations of NITs." This episode continues for another 5 minutes, but for reasons of compactness, we are not including the last part in this conversation. This 2-day FDA workshop on NITs was a seminal event in the development of NASH drugs and diagnostics. The Tsunami team was delighted to attend and thoroughly enjoyed these 3 conversations with leading participants. I anticipate you might as well, so just sit back, listen, learn, enjoy. When you're done, join the dialogue on our LinkedIn discussion group. Let me step back a little bit. First of all, let me say that I agree with everything that all you guys have said so far in terms of how the world works. Okay. But the thing I have in the back of my head is the way a social psychologist wins a Nobel Prize in economics is by proving that we're not averse to risk, we're averse to loss. That people will take tremendous risks to avoid losing things. And then the question becomes, how do you define loss and how do you define big loss? We define loss in all the ways you've been talking about. But part of the reality of government and bureaucracy is they define it very differently. One of the conversations we've had today is with Veronica Miller, who obviously lives close to this from a slightly different perspective. One of the things that had struck me was they spent the first session rehashing Aduhelm and Makina and rosiglitazone, right? So they want to explain why they don't get things wrong, because when they do get things wrong and there's public reaction, it's catastrophic. So my suspicion is that we're going to need to prove a tougher case to them than we would have to prove to ourselves or other academic colleagues. And in that regard, I guess a couple of things that came up yesterday, I think. First of all, one is this question of if the path to regression is not the same as the path to progression, then what does that say about NITs? And more generally, the more you ask the agency, or really any regulatory body, to consider different solutions to different questions depending upon the specifics of the drugs, well, we live in that space, right? But they don't. So I'm a little concerned that if we make it too complicated, the innate fear of making a change and getting it wrong will dominate what do we do. If the goal was to make the first step simple, what would we lean on?
Naim Alkhouri.
No, I mean, I totally understand the concerns. I think, what they realize is whatever pathway we've had, it was a breakthrough. And I really commend them on actually providing us to a drug approval pathway. I think this is what started this whole NASH drug development field, and that's why we're here today. So I think what they did in 2013 was really a big step, and probably they didn't have all the information they needed at the time, but they felt that they needed to act. But we've learned so much about the impact of this pathway on patients and drug development today. So I think it is time to change. Are we in a place to say we know everything we need to know? No, I don't think so. I think there's a lot that we need to learn. But I'll tell you my math, Roger. In our clinic, so we're a hepatology clinic. So by the time the patient is coming to us, this is great because at least they know they have a liver problem. But I see 100 patients with NASH. and only 5 of them will potentially screen for clinical trials, right? So only 5 out of 100, this is in a hepatology clinic. And then when we do all the inclusion-exclusion criteria biopsy, only 1 or 2 end up actually enrolling in a trial. So when you take this to the general population, I mean, this would be probably 1 in 1,000 patients with fatty liver. And these are not the statistics that we have shown over and over. If you remember in our, prospective study, myself and Stephen, we showed that the general population, 14% have NASH and 6% have significant fibrosis. With the current pathway, we turned the most common form of chronic liver disease into a unicorn disease that only exists in, 1 in 1,000 individuals. And just this is not the case. So I just don't want to be in a situation where it's like paralysis by analysis when you have actually a bad pathway that's really not reflective of the patient population that we're trying to target. Every surrogate has issues. Everything can be affected by potentially other disease states. And that's why we're insisting on combination NITs.
Jörn Schattenberg.
I share some of the skepticism Roger mentioned, and however, I do see how the FDA wanted to help the patients and the field to move this forward. So the one comment I thought about for a little bit was the FDA statistician's name. Not sure you realized her. She in the end said, well, just enroll 100 million people and do a full outcome trial. And it sounds like a very straightforward shot, but the problem of, the way trials are currently run, that's impossible. And it negates a lot of the troubles we're going through. So I wasn't sure how to place that. How did that sound to you?
Yeah, I mean, in cirrhosis, we are at that point now, and we are, able to enroll patients potentially without a biopsy. I think we have confidence now in NITs to diagnose cirrhosis. But as you said, once you have F2 and F3, I mean, what are the outcomes? Are we talking about decompensation, ascites, HCC? I mean, that will take a very, very long time, right? And we've seen this even in cirrhosis trials. If you're not including patients with portal hypertension, the event rate is about 3, maybe if you're lucky, 5% per year. So let alone taking patients with F2. And then to do it without biopsy and again, enroll 10,000 people and wait and see how many will develop cirrhosis is just not feasible at this point. So I agree again, in a perfect world where cost is not an issue, sure. I mean, the MRIs we do are not also cheap. So that's another thing to factor in. Yeah, so I don't think that's realistic. I mean, it'll be nice to do, but I don't think it's feasible or possible.
Naim, one of the most compelling things that was shown yesterday, although it wasn't covered particularly, but something we all know, is that it isn't until you get to F4 that liver-related mortality becomes a leading cause of death. If F0 through F3, you have significant impact on mortality, but it's not necessarily liver mortality per se, which to me, by the way, is the best argument not to do your million-person 10-year trial. If you had enough money to do that trial, how many people would die of coronary diseases and other metabolic-related diseases while you were waiting to see if they develop cirrhosis or not?
Yeah, and to be honest with you, Roger, we are so focused on liver-related outcomes and hepatic efficacy endpoints, but we need to also potentially get beyond this and think about cardiovascular outcomes, metabolic syndrome, improvement in each component. Obviously, we look at overall mortality, but there are ways to actually think outside the box and come up with different pathways. But at this point, the FDA made it very clear that to approve a NASH drug, it has to show hepatic efficacy. But again, this is probably not the right way to think about this systemic disease. I think we need to potentially go back to the drawing board and figure out a different way to design trials focusing on overall mortality, cardiovascular, liver, chronic kidney disease, progression to type 2 diabetes and non-diabetics, progression to CKD in people without CKD at baseline. These are all meaningful outcomes.
Louise Campbell.
Well, maybe there's the trial that needs to be written that you've just described. We need a prospective study using non-invasive technologies for metabolic health that include a large number of these ones that we've discussed for the last couple of days, and use lifestyle rather than anything else, and look at what that longitudinal study does. Now, we see lots of longitudinal studies like that in vitamins. They did a 10-year study with 10,000 people on multivitamins, 10,000 not, and ultimately all it did was deliver what we suspected it was going to deliver, expensive poo from one group versus the same health metrics in another. And we've not seen that longitudinal study that looks at the effect of poor liver health and measure it in mental health, because we know that one of the biggest issues with metabolic liver disease is this depression, clouded thinking. People just don't have the energy, and we know that when we defat the liver, mental health is something that improves quickly. So maybe by missing all of those parameters in the past, we've not done ourselves justice, but we're not looking at drugs for that. We're actually looking at monitoring the liver's impact on the body as a whole. Maybe that's what we need to go back to, looking at what happens when the liver either improves or deteriorates. Roger is right. The FDA, like any psychology, you are affected by your future decisions by the results of your past. And it's very difficult to get beyond that. And I would rather go into the future making the wrong decisions for the right reasons, evidence-based, than actually make the wrong decision for the right reasons and use a surrogate marker that has a poor outcome and a risk of harming individuals. And we have seen lots of drugs go forward that were put there for the wrong reasons, even with good evidence. So I think at the time, so there is a balancing act, but psychology is we don't take that step forward because we're affected by our past experiences. But this meeting shows that maybe they will, and at least consider it.
I love this, Louise. I'm going to start using this. So making the wrong decision for the right reason.
Making the right decision with some of the wrong reasons, because they would say using NITs is not the right one. Whereas we've made some drugs that were approved, right drugs, but with the wrong decisions behind them. I think have I clarified that better? Right decisions, wrong reasons. Wrong reasons, right decision. Yes.
Depends on your risk affinity, right? And obviously the side of the table you sit on. But in generally, I agree.
If the agency is looking to take next steps, and the next steps certainly aren't going to be as big as everything that we know, where do you, is your sense of, based on what you heard yesterday, where do you think the next steps are most likely to come?
There's a lot of data that's being generated as part of the phase 3 clinical trials with a lot of NIT data. These patients are being followed long term. The FDA will have also more data to look at in the near future. So that's good news. We're going to also have a better understanding about the risk of outcomes in these clinical trials and how many will progress to cirrhosis. So we can also start thinking in a more informed way about the sample size if we want to do outcomes trials. I think the prognostic data with the biomarkers is becoming stronger every day, especially for the fibrosis biomarkers. And I think the combination NIT field is also maturing, and we are having a better understanding about what it means to have improvement in PDFF and ALT, for example. And we've seen the odds ratios or the hazard ratios with the combination being better than each test alone. I cannot wait to see more with things like MAST and MeFib, and looking even at fast changes, and also changing the categories of these NITs going from high risk to medium or low risk. So I think a lot of data is being generated now, which is good for the field. The way I would love to see it evolving is to have an admission that, hey, you know, what we have today has caused so many problems and is really not a great pathway, that it is time to provide an alternative pathway. And I would like to maintain the status quo, but just have another pathway based on NITs. That would be a dream come true for me, and I know for many of my friends like Jörn and other experts in drug development. So again, to reiterate, all we're asking for, do not get rid of liver biopsy today. I think it's still a viable pathway for some drugs. Just give us one alternative pathway based on combination NITs and taking into account the mechanism of action of the drug. And this will be pre-specified a priori to the trial, obviously. And I think we can, by doing this, decrease screen failure rates, be more inclusive in clinical trials, generate data from trials that will actually be similar to what we see in the real world. And I'm pretty confident that will actually help our patients long term. And at the end of the day, we share the same concerns with the FDA. I mean, I don't want to have drugs that don't work for outcomes at the end of the day. I don't want to medicalize everyone with fatty liver disease and say you need to be on pharmacologic treatment. These are the last things we want to do as physicians. We just want to have a viable pathway that will help these patients, with the end insight is that you need to show improvement in outcomes.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingnash.com. We'll be back next week with coverage of EASL's SLD Summit. Until then, stay safe, surf on, and we'll see you soon on the podcast. Bye-bye now.